Hello and welcome again to another edition of Lost in Science across Australia on the Community Radio Network and through your various podcasting services. And should I maybe say that this week we are Lost in Science Week? Because it is Science Week.
It is. Happy Science Week, everyone. It's the best week of the year. Well, the most sciencey week of the year, anyway.
Depending who you are. Some people probably have more sciencey weeks, but they probably are full-time scientists. So
Yes.
for everyone else, here we are in Science Week.
It's time to celebrate some science.
Yeah.
I mean, we do that most weeks, but really everyone else celebrates some science.
Yeah, the nation. We get the nation to celebrate science.
That's right.
And you are, speaking of the nation, who is made up of humans,
Yes.
who were you talking to this week?
Well, I was talking to a human.
A human?
Dr. Chris Gingell from the Murdoch Children's Research Institute. He's a bioethicist, and he's actually going to be presenting at a National Science Week event called Humans 2.0, which takes researchers from all over and asks them to speculate on what the future of research and the future of humans is going to look like.
Like
So,
evolution-wise?
evolution-wise, the future of antibiotics and medicines, of drugs, and the chemical enhancements,
The singularity?
augmented realities, and where we're going to be with AI and the singularity.
Yeah.
All those sorts of crazy things. The future of gardening as well.
The future of gardening.
Yes,
It'll be
and-
robots, right? It's
Robots
all going to be
gardening.
robots.
Well, you'll just have to come and have a listen. So Chris is going to be talking a little bit about his research, because he looks at some really interesting ethics around bioediting, especially when you're looking at things like CRISPR and the ethics of changing embryos' genetics and their germline into future generations. But then we're also going to be talking about Humans 2.0.
That is really interesting, though, because the thought of that you're not just responsible for the individuals that create through gene editing, but you're responsible for all of the subsequent generations as
Yeah,
well.
indefinitely.
Amazing.
It is incredible, isn't it?
Yeah.
It's big, big ideas.
Huge
Which
ideas.
is, during National Science Week, you want to be talking about big ideas.
We do want to be talking about big ideas. And speaking of big ideas, what's your big idea, Chris?
Well, I mean, you can go and listen to people, obviously scientists, talk about their work and their research, or you can do some research yourself.
That's a great idea. That is a great big idea.
Yes, because every year in Science Week there is a citizen science project, and this year it's one where you can take part in helping research coral and other things on the Great Barrier Reef. I can't guarantee that the government will give you $400 million for doing this research. However, look, it's still worth doing anyway.
This is the thing about Science Week, is that it is about getting everyone involved in science. And there's citizen science projects all over the place. And there's Science Week events all over the place.
All around the country.
All around the country. If you go to the Science Week website, which is scienceweek.net.au, but basically if you Google or use your favourite search engine
You can Bing it if you want.
Yeah.
You can ask Jeeves.
However you find it, if you put Science Week Australia into a search engine, you will end up on the scienceweek.net.au page. And if you if you go to the find events section of their website, you can put in your postcode and it will pop up all of the events around you. One that I saw, which sounded amazing, but it's a little bit out of my reach this week, is there's an event in Broome where they're basically teaching people how to take DNA samples from soil so they can send them in to university science labs and they can figure out what microbes are in soil all around Western Australia.
Oh,
That's
wow.
pretty cool. Yeah.
That is really cool. It's
That
like,
is very cool.
You go and learn, and they say, here's how you take a soil sample, and here's how you preserve it and make sure it all stays alive, and then send it in, and they can figure out, map all of the microbes in Western Australia. How cool is that?
Oh, my goodness.
Amazing.
Wow. I want to go to Broome for Science Week now.
Yeah. Oh, well, next year, I think. I'll try
year.
for next year.
Yeah.
Do a report from there. But there's also, there's science institutions all over Australia. There's observatories all over Australia, and one of them in Ballarat is asking for, mostly for students, I guess, to figure out what would be their top priorities if they were crash landed on Mars. So if they were forced to survive on Mars, what would they have to do? And it's actually a competition. So you bring in your entry of how you would, how you would cope, and you can sort of put it in. But they have a massive telescope where you can look through and have a look at Mars. So you can
get some inspiration.
Yeah.
Martian inspiration
Plan ahead.
while you are planning.
Isn't Ballarat where they make Mars bars, the chocolate bars, as well? Is this like some sort of tie-in?
Maybe it is. Maybe the prize is just boxes of Mars bars.
Yeah, maybe. Maybe that's what you get to look at.
Hopefully, they're also doing some. They're actually looking at the red planet.
Yeah.
Well, there are some other. I mean, Science Week is so great because there are so many grassroots events that are happening in libraries and communities and schools all across Australia. But it's also a chance for people to get on the road and do and tour around Australia with some of their great ideas. And one thing that's happening is the Great Debate, which is the greatest discovery ever made. So in Adelaide, Brisbane, Melbourne and Sydney, to celebrate National Science Week, they are going to have events happening all over Australia looking at the question, what is the greatest discovery ever made?
So that could be a different discovery in Adelaide and in Melbourne and Brisbane and Sydney?
Yeah, well, they've got 8 scientists and comedians and, different people who enjoy science to talk about their greatest discovery. So they will definitely be different greatest discoveries being debated at every different event in every
city.
So
you could follow them, but you could follow them around if you wanted to get all the full set and find out all the discoveries. You could do a tour like a Grateful Dead style.
A science debate groupie. Yeah.
Okay.
I think that would suit you, Chris.
Yeah, I think it would.
And look, it's not all serious science too. If you're in Tasmania, Lawrence Leung is running around the whole country. He's going to be in Franklin and Hobart and Launceston doing shows where he stands up for science, and he's got special guests at every show, talking about cool science things, and obviously making jokes about them as well, I assume.
You know what? You could else is a really good fun Science Week event too.
What's that, Chris?
If you're in Melbourne, you can get along to the Lost in Science Trivia Night, which is the annual biggest highlight of Science Week. I just want to say, it's this year it will be on Monday, the 13th of August.
That's right.
That is right, at the Birmingham Hotel in Collingwood, Smith Street, Collingwood.
Yeah, and we will all be there.
That's right, yeah.
And we'll be delivering some science questions. And I'll also be throwing out some questions on Twitter. So please follow us at Lost in Sci 1.
At Lost in Science 1.
At Lost in Science 1 to follow along on Twitter.
Excellent.
All right, so I think we better get on to talking about Humans 2.0.
You may have heard of the gene editing technology CRISPR and how it promises to be the silver bullet to fix genetic diseases by targeting mutations, not only in plants and animals, but in humans as well. But like every new technology, there is a debate around the ethics of gene editing and what the future holds for this technology. Now, to lead us through the debate, our guest today is Dr. Chris Gyngell, Research Fellow in Biomedical Ethics at Murdoch Children's Research Institute at the University of Melbourne. Chris, welcome to Lost in Science.
Thank you, Claire.
Now, Chris, you've got a pretty great job title, Research Fellow in Biomedical Ethics. Can you talk us through what that means?
Yeah, so essentially I do a lot of academic research on the ethical implications of biotechnologies. So a lot of that is now, as you just discussed, the CRISPR-Cas9 gene editing system. But I also do the ethical issues of other assisted reproduction technologies and, other things that come up. You never know quite what's going to come up in science, and it's interesting to be able to look at the ethical issues as they arise.
Now, a special research interest of yours is CRISPR.
Yep.
which a lot of people might know. In my understanding of CRISPR, it's sort of, or any sort of human germline gene editing, you're sort of taking a sperm or an egg or an embryo and changing the DNA so that all future offspring carry that change.
That's
yes?
right.
Okay.
Yeah.
But CRISPR's sort of new technology.
Yeah, so scientists have been able to modify the genetic makeup of species for decades now. So I remember when I was growing up, I'd see on the news glow-in-the-dark mice and things.
Yeah, of course.
And those was transgenic mice. So I've been able to do that for a while, but those early techniques were really difficult and hard to use. They would use viruses to go in and modify the DNA sequences.
Right.
And they were very imprecise. You'd make lots of different changes to the organism. And so essentially you had to use lots of animals to get the change that you want.
So it was a matter of doing sort of like a shotgun approach and then finding the one edit that you wanted and then going down that line.
Exactly. And often actually what you want to do, you do the shotgun approach, you find the one that you want, and then you breed them together kind of thing. So then you've got this little population with the edit.
Not very exact.
Not very exact, and obviously limited application in humans. So what happened about 6 years ago is a lab in Berkeley run by Jennifer Doudna and Emmanuel Charpentier discovered this molecule called the CRISPR-Cas9 molecule. Basically, this is two components. It's the CRISPR component, which is like a little guide molecule of RNA, which can attach to a specific DNA sequence, and a Cas9 molecule, which is like a pair of molecular scissors. So once the CRISPR molecule binds to a specific sequence, the Cas9 molecule cuts it. And they discovered that they could manipulate this molecule in the lab so it could target any DNA sequence that they want. And then they found that once you've cut the DNA sequence there, you can recruit the cell's own DNA repair mechanisms to add or delete or to alter the genetic sequence there.
Right. So this is no longer you need a specific part of the DNA to insert something. This can be used anywhere on the DNA.
Yeah, essentially, virtually anywhere on the DNA, and you can target it yourself in your lab. So you can say, I want to target this area of DNA, and you make your little guide RNA, and you put it into the cell, and away you go.
Away you go.
Yeah.
Yeah. So in terms of where we are with research on human embryos or human germlines and modifying those, where is the science currently up to?
Yeah, so there's been a few studies. So in 2015, a lab based in China became the first lab to use this technology on human embryos.
That was a while ago.
It was a while ago, and it was very controversial at the time. So at that time, there was a lot of pushback in the scientific community. Both two leading scientific journals, Nature and Science, ran opinion pieces which basically called for this research to be stopped. They thought it was unethical to be pursuing this line of research.
That's a pretty hardline stance for them to take.
It was, and I thought it was a very reactionary stance at that time. Often these studies were done entirely in petri dishes, in embryos that were never going to be implanted into a woman. So it was just basic research. So it's really, I thought it was difficult to sort of say that they were clearly unethical. And since then, we've seen a really sort of softening of the approach to genome editing, and more studies have been conducted in the UK and in America as well, in the USA. And we've slowly started to see more sort of scientific groups and leading scientific bodies sort of endorse this technology for research purposes now.
And what would be some of the sort of like great outcomes that these research bodies are pursuing with this technology in terms of disease eradication?
Yeah, so the most basic thing that they use in it, that they can use it for research now, is actually to study human development. So we understand very little about how humans go from this single cell to this complex multicellular organism. So we don't know what type of mechanisms are involved there. And this is important because humans have a very high rate of spontaneous abortion. So only about a third of fertilized eggs survive the first trimester of pregnancy, and we don't really understand why that is. And also IVF success rates are traditionally quite low. And that may be because we don't really understand what's going on at those very early stages of development. So what they can do with this technology is alter particular genes in an embryo and then watch it grow, and then that gives them a clue about, oh, well, that mechanism, that gene's doing something really important in that early stage.
That sort of research is looking at the embryos, and then that would not go on to then become...
That's right. So all this research is done, there's generally a worldwide consensus that when you do research on embryos, it can only be done to 14 days.
Okay.
So nothing above 14 days is considered ethical or legal in most jurisdictions, yeah. So we're talking about the very early stages of development.
Wow. Yeah. And what's the current sort of philosophical debate about these sorts of technologies in Australia specifically? Are we behind the rest of the world? Are we somewhere in the middle?
In Australia, it's very unlikely that you can do that research. A lot of people interpret the law as saying that those type of studies are illegal in Australia, and it's certainly not been done in Australia. So there's a lot of debate now whether we need to change those laws to allow that type of research. My own personal view is that we should. I think it's really hard to see how that research can be harmful, particularly in jurisdictions like Australia where you've got, good oversight, good regulations, and it's generally, considered worldwide a good place to conduct scientific research.
In your opinion, what does the future of gene editing on embryos look like?
Yeah, so I've talked about what you can do in research terms, but obviously people are much more concerned about not just doing it for research, but for using it for reproduction. So initially you could do that to treat genetic diseases. So a lot of genetic diseases are caused by one defective gene or one mutation, such as Tay-Sachs syndrome or cystic fibrosis. With gene editing, you could potentially find those genes in an embryo and correct them. So it would be a one-hit cure kind of scenario. And that sounds really good. And I think, obviously, we like to prevent disease. It's generally seen as an ethically good thing to do. But you get into more tricky things about, well, how far do we go? What if we want to change the eye colour of a child, or their height, or their intelligence, or their sexual orientation? So you get into these really sort of tricky areas quite quickly.
When it stops being life-threatening, it then just turns into something that the parents have a choice about. That's extremely tricky.
It is, yeah. So there's a few tricky things. So one is like, who should decide? So who should make those decisions about what type of genes and traits a child should have? Some groups of philosophers think it should be up to the parents. They endorse a principle of sort of reproductive freedom, saying that, well, I should be free to go and live my life how I want, and the government or laws should only stop me doing what I want when I'm at risk of harming someone. So if I'm doing something to an embryo that's not going to harm it, say we don't think changing an eye colour is necessarily a harmful thing to do to them, then I should be free to do that, and I should have the right to do that. Against that, you have people saying we've got to think more than just the individual here and think about the collective interest in these technologies and the risk to changing the sort of child-parent relationship. Will children start to be seen as sort of objects that we're using rather than sort of an ends in themselves? People also worry about the sort of social consequences of maybe only some people will be able to afford these technologies, generally the rich, and maybe they'll use it to give themselves even further biological advantages, and so it will worsen inequalities.
Certainly, and the agency of the unborn child, they then grow up to realise that they've been designed in a certain way by their parents.
Yeah, that's right. So another group of concerns is about, in general, for medical technologies like this, informed consent is a really sort of strong medical principle. You obviously can't get consent from an embryo, so therefore perhaps we shouldn't do it. Personally, I don't think those are the strongest argument, because there's a lot of things we do to infants which we can't get the consent for. And we don't get consent to do open heart surgery on a 2-week-old baby, but we do it because it might be in their best interest. But of course, that comes back to what you said before about the distinction between something life-saving and something that's just done as a preference.
Where are these debates currently happening? Are they happening in a rigorous way? Are we examining this as much as we should be? Are we educated enough?
Yeah, so look, Australia's a little bit behind. There's a lot of rigorous debate going on in the United Kingdom at the moment. So an influential independent body called the Nuffield Council of Bioethics 2 weeks ago released their report on genome editing and human reproduction. I actually provided input into that report. I was sort of interviewed there while I was in the UK. And that's a very rigorous report. And I think they've got quite a good approach, which is they're not actually ruling anything in or out at this stage. Rather, they're sort of trying to get the underlying ethical principles which guide the development of the technology. And what they sort of came up with is we need to consider the well-being of any children that are produced. We need to consider the effects on society and make sure we don't marginalise any social groups. And we also need to consider the effects of further generations far, far, far into the future.
Well, that sounds like a very good starting point.
Yeah, I thought so too.
Now, Chris, you're actually going to be presenting your ideas at a National Science Week event, Humans 2.0. It's in Melbourne on Wednesday, August 15. What can people expect from that?
It's going to be a really, really exciting event. So I'm obviously going to be talking about genetic engineering and how that might affect future generations, or how, if we don't do any genetic engineering, how that might affect very future generations as well. But you have seemingly a lot of exciting things to do. There's a lot of people doing immersive experiences and virtual reality and robotics. And yeah, I think it's going to be really fun.
So the event is all about the future of the human race. So we're not talking about necessarily, I mean, you know, some of it is grounded in research that's taking place now, but we're projecting ourselves into the future.
That's right. So it's really exercising our imagination of what will the human species look like in the very far future. And, you know, I think what's really interesting is you've got all these different perspectives and you're going to have some quite different pictures, I think, presented on that night.
Absolutely. Well, I'm going to be going along and I can't wait to hear your presentation. Chris, thank you so much for coming in to Lost in Science today. And if you are in Melbourne, make sure you head to Humans 2.0 on Wednesday, August 15, and you can find details at scienceweek.net.au.
Across Australia on the Community Radio Network, you're listening to Lost in Science.
Yes, you're listening to Lost in Science Week. And as I said in the introduction, the government may be handing out funding to Great Barrier Reef projects of dubious value.
I've got a Great Barrier Reef project. Can they give me some money? I just want to go on one of those glass bottom boats and look at all of the Great Barrier Reef. Will they give me money for that? I'll write something down as I was there. I'll record some data.
Well, that is strikingly similar to the project that I'm going to talk about, Stu, which is
good.
one where you can actually take part in a project for part of Science Week to actually help the Great Barrier Reef. You probably won't get millions of dollars for it, though. Unfortunately, yeah.
Will I get a glass bottom boat? That's the most important part of my project.
Well, not.
You will get the satisfaction that you are, you know,
Contributing to science.
contributing to science and the
Well,
further development
that's more
of
than enough, really, isn't it?
the scientific knowledge base.
Yeah.
That's right. You can get a glass bottom on your car or something. You can drive around looking at the road. Anyway, no. So this project is called Virtual Reef. It is the citizen science project for National Science Week for 2018. Flagship.
Yep.
Every year they have one. It's usually promoted by the ABC. We don't hold any grudges, though. You know, we're fine with the ABC.
We're all together in the
together.
non-commercial.
That's correct. That's
broadcasting
correct.
space.
They're our colleagues. Last year was a smartphone survey. I don't know, it was called the smartphone survey.
This one sounds much more interesting.
Yeah.
Well, many of the projects are similar to this kind of one, which is basically kind of where you have a whole lot of data that you need an army of humans to process. So in 2016, they had Wildlife Spotter, which used photos from trail cams. You basically had to say, there's a wombat. And
So what, you just sit there and watch footage and wait? And
They're
if a wombat
photos.
walks
They'll be
past-
photos. They'll be
okay.
show you photos and you go, is there an animal in this photo? What is it?
Okay.
Kind of thing. In 2015,
There was a sleep one as well, was there?
that was quite a few years ago. Yeah,
right.
that wasn't this same kind of thing. You didn't look at people sleeping and go, yep, sleep, yep, awake. But they had one in 2015 that was called Galaxy Explorer, pretty self-explanatory.
yes.
Yeah, you're looking at
Yes.
pictures of galaxies.
Okay.
2014 was one called Weather Detective, which basically looking at climate records in old captain's logbooks. 2013, there was one actually called Explore the Seafloor, which is specifically about mapping kelp and sea urchin populations. But, you may not be into kelp and sea urchin. I don't know your taste. But this one is actually this year is about the Great Barrier Reef. And so it's looking at things like the extent of coral cover over the reef. We know that the reef is under threat, obviously, mostly from climate change, as well as, other factors that are going on. But, what is the damage? What is the extent of the damage? What is it likely to be? How can we model that? We need data to be able to make these predictions to understand what's going on. And you can send teams out to do surveys, like Stu. You can pay for Stu to go out in his glass-bottom boat. But it takes a lot of money and a lot of stews to get a good coverage of the reef. So the idea here is basically you just have lots and lots of photos of the reef, and then you get people to sift through them. So it's the kind of thing that's harder to solve with money. You just need people to tackle that. So yeah, because there's a limit to what money can buy. You need human brain power.
We don't have coral spotting AIs yet that are good enough to figure it out. Is that the point?
Unfortunately not.
We have that Crown of Thorns spotting AI, which machine learned how to spot a crown of thorns and then has a deadly injection that when it finds a crown of thorns, it injects it.
Is it a robot that goes out on the reef?
Yes, it
Yeah.
is. Don't you remember that?
Didn't they call it the Terminator robot or something?
It's an incredible robot. I did a story a couple of years about it.
Of course I remember it then.
Can't believe you don't remember my stories, Chris.
Anyway, this one has got... I'll tell you how it works. So it's being run by statistical scientists from the Queensland University of Technology. The database, and I had a look at it, was over 68,000 photos. Sounds like a lot, but we'll get on to that. And what you do is you log on, you create an account, and then you tell the computer what you see in the photo. So it puts up a little photo of the reef, and it puts 15 small circles randomly placed over the photo, and you have to say what's in each of those circles. So you're not looking at describing the whole thing. It doesn't like put you just a picture of the reef and you go and you write a little text thing going, I see vistas of coral and waving sea fronds, whatever, those sort of things. You goes, okay, here's a circle.
Quantify it.
So that's the idea. It's to get quantified data. You're basically getting an image, a qualitative image, and you're basically going, we're trying to quantify this. So so do things like the extent of coral cover. You're actually counting the amount of coral effectively. So it has these 15 circles, and you say what's in those. So you've got like hard coral, soft coral, sand, water, algae,
Fish.
There's an other option.
Sea turtle.
Yeah.
That'd be a good photo, wouldn't it?
So, and the same photo is going to be shown to multiple people because they'll put the circles in different spots. But the idea is you also
need to verify
you also get processing. Like the computer has to then make sense of it all. Because I got to say, I logged on and it was mostly coral from what I could tell. But I'm not an expert, and I'm doing what I think it is, and someone else may differ. There is training videos you can watch. It'll give you some idea. But, like the hard coral, soft coral, you can't touch it. That's what I'm saying.
So do they cross-check these results, or are they matching sort of similar, like 20 people see the same photo and they figure out what every person...
I don't know how the algorithm works, but they are doing that. They are essentially, they're combining all this information to get... to get an estimate of what's there. And the main thing they're looking at is coral cover. So really that's the thing they're focusing on, is you're telling what's coral and what's not coral. So I looked at a few pictures, and some had basically a whole lot of coral with maybe some little anemones and things there. And other ones was mostly sand. And it's pretty easy to spot the bits that weren't sand.
Right.
Yeah, so it's pretty straightforward.
So do we know what's going to happen to this information once we've all given our time? Are they going to come back to us with the research paper with all our names on it?
With the results?
Well, presumably so. It is about primarily about modelling what's going to happen to the reef in the future. And so they have got a bit of information on their website showing how that the more data you have, the more accurate your models can be, and how you can fill in the gaps. Because, they have, like I said, surveys that they've done, and there's lots of gaps between those. So this is trying to fill in those gaps. But it really is about, trying to predict what will happen under climatic conditions, but also under, say, if there's a cyclone comes through, what will the effect of that cyclone be? So, I don't know. They haven't said
bleaching event.
Yeah, they haven't said specifically what the papers they'll produce are. I imagine there usually are papers published for these things, and they don't credit every person, unfortunately. You don't get like a paper in your name. Although the Didn't the recent Stargazing Live, they discovered...
I think they discovered some planets that were orbiting a distant star.
They discovered a new solar system.
And then the person and the citizen scientist who discovered it was credited in the paper.
Along with Julie Zamiro.
Along with Julie Zamira.
That's
Who
right.
was hosting the show. Yeah.
Yep. So, yeah, they're going to predict this. They're trying to basically remove the gaps within their data. Now, 68,000 photos, I said. That sounds like a lot of photos, but the Great Barrier Reef covers 350,000 square kilometres, and these photos are actually quite zoomed in. So they do need a lot more photos. So people are actually invited to upload their own photos and add to the database so they can get more coverage of the reef and try to fill in the gaps. So, yeah, it's the more data you get in, the more, like I said, the more accurate the model, the better the prediction. So look, it's a very easy project. You basically you're looking at pictures of the reef, and you get to contribute to science and to the future of the reef. What more could you ask for? $400 million, obviously. But apart from that, the feeling of a job well done. If you want to find out more, you can go to virtualreef.org.au to find out more about the project and to register and take part.
We've run out of time to get lost in Science Week this week on the show.
The week goes on.
It does. And wherever you are in Australia, there is some science happening around you that you can even join in on. You should get onto the Science Week webpage and have a look at what is happening in your neighbourhood, wherever you may happen to be.
That is scienceweek.net.au.
Yeah, that is correct.
But if you are in the Melbourne region, then please come down to the Lost in Science trivia for National Science Week, and also as a fundraiser for 3CR. We would love to see you down there. We'll kick off around 7:30, but come down to 6:30 for hangouts and maybe a bite to eat.
And meet the stars of Lost in Science. That's us.
Oh, that's us.
Yep,
Oh,
yep.
Yeah, we better make sure we're there too.
Yep.
Yeah. That is the 13th of August from 6:30. And you can check out all the details on our Facebook page. You can find us at Lost in Science and 3CR on Facebook.
It's $15 for 3CR subscribers and $20 for people who are not yet subscribers.
Why wouldn't you?
So, thanks for joining us on Lost in Science. Lost in Science is recorded at the studios of 3CR in Melbourne and broadcast across Australia on the Community Radio Network with the financial assistance of the Community Broadcasting Foundation. You can, as we mentioned, reach us on Facebook to give us your commiserations for not making it to trivia, or you can email us, or you can just tune in once again next week when the Lost in Science team all get together and get Lost in Science!
Thanks for listening to a 3CR podcast. 3CR is an independent community radio station based in Melbourne, Australia. We need your financial support to keep going. Go to www.3cr.org.au for more information and to donate online.